Hello, it's a pleasure to talk to you today about our work in basic and translational integrative medicine. Before I came to Osher, the Brigham, and Harvard Medical School, I worked in the pharmaceutical industry in drug development, where it was critical to manufacture the same drug the same way every time. While this guaranteed safety, it did not guarantee efficacy, and I became fascinated by the wide range of responses we saw to one treatment. Precision medicine is designed to address this very variability in treatment response. While current methods look for genomic subsets of patients who respond to a treatment, it doesn't really look at the subset of patients that respond to placebo treatments. Thus, pharmacogenomics and precision medicine are missing what Ted Kapchuk and others have argued is one of the most powerful drivers of treatment response. What's more, when you consider that systems, pharmacogenomics, and placebo effects interact in clinical trials, the matter is even more complex than we ever thought. In traditional clinical trials, where placebos are used as controls, we randomize patients to a drug or a placebo treatment and compare the difference in the outcomes in the treatment. This difference is considered to be the drug effect, and it's how we determine the efficacy of a drug. But more and more, in randomized clinical trials, what we're seeing is there's no difference between the drug and placebo. And so in clinical trials for Alzheimer's, Parkinson's disease, depression, or IBS, we're finding that drugs are not able to beat placebos, and thus millions to billions of dollars are being wasted and patients are being left without much needed treatments. The idea behind precision medicine is that it is possible to identify subpopulations for whom a drug is efficacious, but the problem with precision medicine is it does not often consider that there might be populations that also benefit from placebo that may or may not be confounding the effects of the drug. And so our mission was to use pharmacogenomics to see if we could identify a genetic signature of a placebo responder and compare that to the genetic signature of a drug responder. When we looked for genes that were associated with the placebo response, we zoomed in on a group of genes we called the placebome, and here is an example of one such gene, catecholomethyltransferase, or COMT. The COMT gene, when looked at in the context of an irritable bowel syndrome, was found to actually be associated with placebo response, such that there was a subpopulation that responded to placebo and a subpopulation that didn't. And this was true in more than one randomized controlled clinical trial of IBS. And this led us to think about where else could we see an association between the COMT genotype and outcomes in the placebo arm. As we looked throughout the literature, we realized that COMT not only seems to interact with the placebo effect, but it also seems to modify the effect of several drugs. And so we asked our question, what happens when you have a drug and a placebo response that is being perturbed by the same gene? And to give you an example of how this works, let's take a look at the Women's Health Study. The Women's Health Study is a landmark randomized clinical trial that was led by Julie Buring, one of our advisors here in the Osher Center. Under Julie's leadership, the Women's Health Study randomized almost 40,000 women to vitamin E, aspirin, or placebo, and followed them for 10 years and examined the rates of major cardiovascular disease and cancer among these women. There was, to this point, compelling evidence that vitamin E as an antioxidant would be protective from major cardiovascular disease events and for total cancer. But at the end of this trial, it was found that there was no difference between placebo and vitamin E in the rates of major cardiovascular disease and total cancer. So we asked the question, if we looked at COMT genotype, in this population, could we see differences in how the women responded to vitamin E? And that's exactly what we found. In both cancer and cardiovascular disease, we found differential responses to placebo and vitamin E, such that if we were to average the outcomes in each of the treatment arms, you would see no difference. But when you looked at the outcomes by COMT genotype, we began to see that there were significant differences, such that there are populations that benefited from vitamin E. So this became one of the first suggestions that to identify pharmacogenomic candidate genes, it was important to not just look at the outcomes in the drug treatment arm, but to strongly consider what was happening in the placebo arm. And this led us to ask an interesting question. Surely there are other genes that are being influenced or influencing placebo effects, and how many of these other genes are also affecting drug effects? To address this is a complex problem involving myriad genes and drugs, and understanding how they interact and how we might intervene to look for gene-drug placebo interactions. So we're turning to network medicine and systems pharmacogenomics, while at the same time we're very interested in looking for drugs that target the placebo effect. On the right, you can see that we have identified riboflavin and rosmarinic acid as two supplements that interact with COMT and potentially can be used to perturb COMT to potentially boost a placebo response and take a novel approach to conditions like IBS that are very sensitive to placebo effects. Placebos are not only important in clinical trials. Given their striking ameliorative properties, and given that the presence of their opposite, the nocebo effects, can be harmful, we're beginning to examine the impact of the absence of placebo and the presence of nocebo on health equity, and in turn, population and public health. From vaccine to statin hesitancy and resistance, we see that these effects are spreading through an infodemic, and it's incumbent on us to stop their harmful effects. Recently, I've had a wonderful opportunity to synthesize much of what I've learned on placebos in a book published by MIT Press, which is coming out in October. In this book, I make the case that placebos represent a backdoor to our health and wellness, and throughout the years, it has been championed by physicians and manipulated by quacks and hacks. And more we're seeing this in statin and vaccine hesitancy across this country and across the world. And I make the strong case that protecting the back door is the work of us all. Our goal is, in the future, to identify other genes that influence placebo response and fill out what we call the placebome. We hope to look for and find genetic subpopulations that can benefit from supplements using these genes in the placebome. And we also hope to identify supplements and drugs that can target the placebo response so that we can boost it and manipulate it to benefit patients. And we're in the process of developing a clinical trial with quercetin, a drug that binds to COMT and inhibits it. And we want to do this clinical trial to look at pain in IBS. And finally, we are very committed to understanding the impact of placebos and nocebos on patients and health equity and the public health at large. Thank you so much for your attention and your time.